All right, I think we're gonna start now. All right, thank you everyone for joining us today, those of you on Zoom and those on Facebook. It's really exciting to be hosting Professor Franklin Aigbirhio today with us. He'll be talking to us about development and application of PET imaging for dementia research. Just a reminder, this SONA webinar series, the whole idea is to bring in experts from around the world to give a talk about their research in order to inspire other researchers as well as create collaboration opportunities. So before we start, let me give you a brief bio about Franklin. So Franklin obtained a BSc in chemistry from the University of East Anglia in the UK, followed by a PhD from the University of Sussex. He entered the research field of radiochemistry and biomedical imaging on joining the MRC Cyclotron Unit at Imperial College London in 1991. He then joined the Wolfson Brain Imaging Centre, University of Cambridge in 1997 as a founding staff member and helped to establish its PET programs and progressed to co-director of the center in 2016. Presently, as a professor of molecular imaging chemistry, he manages a lab undertaking research programs in developing novel PET imaging probes and methods and their application to preclinical and clinical research in neuroscience. He is the lead for the Dementia-Platform UK Imaging Network, a senior research fellow at Magdalene College Cambridge, and a director of the International Society of Radiopharmaceutical Sciences. It's a huge pleasure to have you with us, Franklin. And before you start, let me just remind our audience that when you have questions, you can just type them into the Q&A box on Zoom. And for those of you on Facebook, just type it as comment. I'll try as much as possible to go back to Facebook during the Q&A session to read your questions. Thank you very much. Franklin, the stage is all yours.
Thank you very much, Mahmoud. It's a real pleasure to be able to present here, and I think, Mahmoud, for this, if this kind of a platform for people who are interested in this subject or what I'm doing as a platform collaboration, I'll be certainly welcome for doing that. So let me just kind of give you the kind of where I'm from. So as I said, I can see from my heading, I'm obviously a professor here at the University of Cambridge, but also I'm also kind of two things, also the lead for Dementia-Platform UK Imaging Network, and I'll kind of come back to that, touch on that kind of later on in my talk, and it really kind of overlaps very much what I'm going to present. So I'm kind of minded that this is kind of around the world sort of thing in terms of this talk, and so it's really, I suppose, kind of set the scene and in terms of where I'm based and working. And kind of based here at obviously based in Cambridge, and just on the south of Cambridge is called the Cambridge Biomedical Campus, which at the core of it is the major regional hospital, Addenbrooke's Hospital. And over the last few years, we really expanded to almost like the largest kind of biomedical research campus in Europe, which of course includes at the Addenbrooke's Hospital, the local hospital for cardiovascular, AstraZeneca, you can probably see their kind of large building there, and the MRC Laboratory for Molecular Biology, maybe people have heard of as well as there. And of course, being part of the University School of Clinical Medicine, in which I'm based. And based here at the Wolfson Brain Imaging Centre itself, which is kind of a little picture here. And again, I'll maybe touch on that in a bit. So I suppose one of my briefs, I think, when I was invited, when I was kind of, how did I get here? And really, I put the kind of title here, Applying Chemistry to Biomedical Research, which is at the core, which I always forget, is what I'm doing. I'm a professor in the Department of Clinical Neurosciences in the School of Medicine. But what I am is chemistry and applying that to my biomedical research. So I had a degree in BSc in chemical science, as Mahmoud said, from University of East Anglia, not actually far from Cambridge. Then I got a DPhil in physical organic chemistry. So really, so far, nothing to do with what I'm doing then. And then I did a postdoctoral in organic metallic chemistry. And without kind of going through all the various steps, I finally ended up here at the Wolfson Brain Imaging Centre, and really far removed from the concept of what I used to do in the lab, I find myself really what was a very much clinically focused research department. The core of the Wolfson Brain Imaging Centre, and it says the brain underpins it, what we do, was very much early aspects of understanding research in acute brain injury, and yes, in sense of actually doing a scan with a person on brain injury very early post-ictus. So this is really where I come from, and that's, you could say, is my journey from chemistry to very much the front end of clinical research. But the core of it, and as I go through in terms of explaining it, is applying chemistry methods and chemistry molecules, different chemistry molecules, to support biomedical research. And I said where we are based is the Wolfson Brain Imaging Centre, and that is a dedicated centre purely focused in terms of applying what the term used as molecular imaging, it's a more kind of a wider term of what to do to background research, primarily on neuroscience, early days on acute brain injury to more stroke, to what we are come through now into a wider areas of neuroscience and non-neuroscience aspects. So what we have, and underpinning our molecular imaging program, is a manual MRI, which clearly most people have kind of heard of, and it's a common techniques in many hospitals. But we have apart from the most standard 3 Tesla in terms of power scanners, we have a 7 Tesla, which has a more ability to kind of more detail in terms of especially in terms of brain imaging. But the core, which I'll expand on, is positron emission tomography, and this is the area which my area of research is, which for many people maybe haven't heard about, is a nuclear medicine type technique that uses radiopharmaceuticals to image and measure brain biochemicals. And in my next scan, I'll kind of bit more touching about it. But here's the actual scanner in the Wolfson. And they've become more sophisticated now. Before, they used to be standalone PET scanner, but as a common theme now, this actually has a combined PET MRI scanner where you can then get the advantages of both technologies. So you have able to scan for MRI for that high resolution structural of the brain. And here it is in high detail, which is the real strength of MRI. But then you can also co-register and at the same time image and obtain the biochemical and less resolution of a PET. And really that becomes a real strength of what we can do now in terms of this technique. So a bit more detail of what is positron emission tomography. The core of it is, in the essence, is as I say, a non-invasive technique. And it's based on using what are very short-lived radioisotopes, which in terms of that, which emit in a unique way of emitting with a positron from their decay. And in that positron, it coincides at a very short distance with the electron, and it emits 2 coincidence gamma rays. And that's a real advantage, a coincidence. And with that ability of coincidence and having what are very sophisticated scanners, high res in terms of sensitivity and resolution, we actually detect this the point of this emission. We can then quantify, due to the fact it's got this coincidence gamma rays, we can actually come down to the point of it. So you will inject a radiopharmaceutical, therefore a drug of any form of chemical, which incorporates a carbon isotope. And of course, in distribution in the brain, we can then get this distribution linked to some pathology, and then indirectly determine that pathology by using this technique. And you can see kind of image which we'll get. In the sense, the resolution doesn't compare well to as well as MRI, but still can be very powerful in a smaller type dedicated scanners for using, say, small animals, 2 millimeters, while for more of our more clinical programs, scanners, which we have just showed you example, 3 to 8 millimeters. But the real strength, and of course, is its high sensitivity, able to go down to the nanomolar pico-level levels to therefore quantify enzymes, receptor in actual quantification. And that is a real advantage of that. So in essence, and of course, as I kind of touched on about animals, also ability, and this is an example of what we've did several years ago in Cambridge. You can use the same kind of tracer to then determine the same pathology and correlate pathology in an animal model with a human model using exactly the same kind of radiopharmaceutical. And in this case, we're using a tracer which maps onto dopamine due to receptors, which tends to be found in striatum in the brain. So we can then correlate, use exactly the same thing to do a study in humans and in terms of animal studies to actually correlate the same pathology using exactly the same technique of the same radiopharmaceutical. And of course, in this case, maybe a different, same kind of scanner, but in this case, optimized for animals. So it's really a powerful technique for translational understanding and going back translation humans to animals and forward as well. So those are the advantages. The challenge of it, and of course, the challenge of it is that, I mentioned short half-lives in terms of the radioisotopes, and they are short. And we in a sense, as so examples, the most commonly used one is carbon-11, a variation of carbon-12, which is a stable organic carbon fins. It's got a half-life of 20 minutes. So every 20 minutes, it decays into half the activity and onwards. Shorter ones, nitrogen-10, but the two ones which we tend to use, we do use oxygen-15, 2 minutes, but even with 2 minutes, that's certainly a challenge, so we can't do too much, but we can still do a bit. But the two key ones, carbon and the variation of fluorine-18, not in a stable form, is fluorine-19, it's got 2 hours. So they are short, but that's the advantage. But of course, being short, in essence, they can't be kind of made and stored. They have to be, in essence, generated on the day and used on the day. So what we have in the Wolfson and major comparable PET centers will have is what this is kind of a cyclotron, which is here. And in terms of, I suppose, to put in context of size, if I was standing there, I'll probably be about there. So it's quite a relatively compact system. This is with the magnet open. Normally this will be shut. And what you can do, starting from, you can generate charged particles through this at high energies. And then in those ports, it will then convert stable material into any of those variations of isotopes. So that's what is kind of starting material, generate any of those on the day, on the site, and then the next step, which is again, this way is a challenge, is get a way to incorporate that isotope into a pharmaceutical, into a drug, into a biochemical of interest. And of course, due to the nature of them being very short half-life, the whole process has to be rapid. And rapid in terms of making what it, in essence, could be a medicine, when I say pharmaceutical, radiopharmaceutical for human use. Process time could be 60 minutes to 2 hours. And you go through the cyclotron generation of the radionuclide, the actual synthesis, therefore getting a means to incorporating the isotope into your drug of interest, purify it, make sure it really represents the radiopharmaceutical of interest, formulate it, put into a form that's acceptable for human use, quality control, confirm it is the compound radiopharmaceutical of interest, it's pure, it's quite the right pH, etc., before it gets applied into humans. And we can make, just to put it in context, I put these figures 0.5 to 3 gigabecquerels, which is a measure of radioactivity. And to put in the context, for a normal human study, we'll probably use about 0.25. So we can make generally more than enough for one patient study. But despite the challenge, over the years, we've built a real power range of different types of radioisotopes. So we can measure cerebral blood flow with a variation of water. we can use oxygen 15 as a measure to the oxygen metabolism. I'll come back to this. Of course, if there's one PET radiotracer you might have heard of, which probably represent, I would say, 95% of clinical application worldwide, UK, US, Europe, is this one fluorodeoxyglucose, a variation of glucose which incorporates a fluorine 18 in terms of the hydroxy group, which gives us this unique ability to measure glucose metabolism in the brain. But there are also other things in terms of targeting enzyme, in this sense, diphenol for monoamine oxidase B, or targeting receptors, dopamine D2 receptor with this compound, actually a failed drug called raclopride. And you can see this 3D rendition of a scan. We actually see with this dope, raclopride, the striatal well defined by this specific uptake of this receptor in this compound in our brain. So this is really the kind of things we can do. But as I said, what I really focus on is really in terms of how a really successful area in terms of bringing on a range of different radiopharmaceuticals to then interrogate a part of a pathology is being neurodegenerative disorders. And I say I come back to, I lead in the UK a network for dementia for applying imaging for dementia research. And obviously many people will know neurodegenerative disorders is the catch-all term for a range of disorders where you get global cognitive decline leading to functional impairment. And of course, there are very many causes and linked with different kind of proteins. Most commonly is Alzheimer's, Parkinson's, vascular dementia, Lewy body dementia, and for the dementias. And of course, it's something that is rising prominently in the UK and other countries around the world. And I think this is the latest figure, about 800,000 in the UK, with numbers set to arrive. And of course, apart from the social burden to the patients, the carers, the economic burden on that. So it's a major thing which and of course, there's at present no actual drug works, even though it's real promising with the recent announcement with Biogen, a potential drug that does more than just deal with the symptoms, that actually targets the pathology. And the core pathology is aspects, in this case, example of Alzheimer's, where you get amyloid plaque, protein deposition, or tau tangled, again, in a form of atomic progression. So this is where, in a sense, we've kind of able to try and use PET imaging to do that. Now, when I used to, a long time ago, do this tour, I used to kind of show this kind of what is considered a cascade hypothesis of Alzheimer's disease. But about working with so many neurobiologists and Alzheimer's, I really do not show this anymore. This kind of almost like a sequence of events that occurs to the dementia. And I think it's quite clear it's certainly more complex than that. So it's probably more appropriate to put it like this in no particular order. The only case I can find, of course, there's neural cell damage and dementia. So it really, in sense of how these different components of pathology are occurred, their timelines and the interactions, this really is where you can actually apply PET to using that to what I say is delineate the components of dementia pathology, image in individual components, and then define their relationships and time course to each other. And this kind of things which we at Cambridge have been doing, establishing, we could say, a portfolio of specific radio tracers that target different pathologies to end up enabled for research into this these new disorders. And this links up with what we also do on the apart from a Cambridge scale, kind of wider UK scale in terms of applying this methodology. So I can't go through maybe if time in terms of what we tend to use. This I mentioned again, fluorodeoxyglucose. It was very much one of the earlier and still the most prominent PET radio tracer for, and what it does, it measures energy use based on glucose metabolism. It goes into glycolysis process, but because of this fluorine 18, which means it can't be phosphorylated a second time, it gets fixed in the cell and gives a real powerful marker of rate of glucose utilization. And of course, that's something which we can apply in terms of understanding, especially comparing the healthy brain to a brain with dementia, in this Alzheimer's brain. And you can see this is, as you said, PET tends to be more lower resolution than you might see in MRI, but you can see a healthy brain with all the high uptake of glucose. And of course, in the brain uses a significant amount of glucose compared to the rest of the body, so it's not often such a sensitive mark as other things I will come through later. But you can see a lower, the kind of darkening of the Alzheimer's, and it's certainly the kind of where you get a kind of lower brain in terms of showing kind of atrophy of the brain, so therefore less in terms of cells available to use the glucose itself. So basically you do see that darker and darker level. So I suppose in this case you get a negative, you get a drop in the use of glucose compared to the healthy cells. So that's a kind of a tool we can use, and it's still maybe not as used as often, but still a very powerful tool. Depending on the parts of the brain that has atrophy, you can distinguish between potentially Alzheimer's from other forms of dementia and such. But it's kind of I wouldn't say a crucial, but it's less in terms of specific to a new generation. But really the breakthrough for this field and in terms of imaging was for the first time to be able to image the actual pathology linked with neurodegeneration, which is the misfolding proteins, which are in a sense toxic and of course have this characteristic common beta sheet character. And the variation from a compound called TIF11T was developed by Bill Clonk and Cohen Chet Mattis years ago, with this thing called PIB, which has a specific and selective imaging of beta amyloid plaques in the brain. And here's that was a Carmel 11 version, and a more commercial available version, which potentially been used for diagnostic in terms of application for Alzheimer's, is one from this for a company from GE. And there's some work which we did at Cambridge. In this case, you can say a typical cause of dementia, and it's actually in Down syndrome, which have very much earlier onset of Alzheimer's. And you can see the uptake, this scale of the uptake of from low uptake to high uptake in this brain of this adult. And you can see the real uptake of this tracer in that, showing there's a high deposition of amyloid plaque. So moving on, and of course, this is in a sense is come even more prominent in more recent years in the recognition of inflammation is something which not necessarily just something that's kind of outcome, but potentially could be an initiator or a aspect, a key for pathology in very new genetic disorders. And that's certainly risen in terms of interest, in terms of understanding the role of this in dementia, and even now, and out of that now, there actually are therapeutics potentially targeting this as an alternative to the standard therapies for dementia. And the way we image this is really by looking at the active microglia, which in a normal brain, healthy brain, are nascent, but when it becomes some kind of inflammatory process, chronic or acute, and of course more chronic in terms of dementia, they come activated in activated form, and they, in this case, they express a receptor called TSPO receptors, and we can use that as a kind of a biomarker, indirect biomarker of activated glia. And in our studies, it's actually confirmed that there's a correlation between the amount of expression of this TSPO, the activated microglia. So we can actually quantify it and relate it to the presence of microglia. And the original PET tracer for that is called PK treatment 95. Again, a compound labeled tagged with a carbon 11 in this point, and then we can get this kind of image which we've done at Cambridge. You can see the real inflammatory process in late stage Alzheimer's. So again, we can actually look at this component in relation to what's going on, and it's a newer form of the trace. And you probably see a common theme. We tend to start off with a carbon-11, but of course its half-life of 20 minutes means it's always a challenge to work. You can always usually just make enough for one person. While with a longer half-life of the fluorine with a 2-hour half-life, then it's certainly more relaxing for everyone to use. You can generate enough from our cyclotron and from the radiopharmacy to then make enough for 2 to 3 patients, which of course makes it a far more economical and also a improved throughput for doing these studies. So on to the next one, I think of course the key next one for terms of toxic proteins, and certainly you have amyloid, which is present of course in Alzheimer's, but a more prominent prevalent misfolding protein is tau, which not only is present in Alzheimer's, but is in a range of various traces of dementias, which are commonly known as tauopathies. And again, it's a misfolding protein, and in this case, it creates in the brain called neurofibril tangles. In terms of Alzheimer's, there are different forms in different pathologies. And the really first radiopharmaceutical that targets this is this one from originally called TO27, AV145, which again in this case is selective. Selectively will only mark neurofibril tangles, and it does not show and doesn't show at all the amyloid. So in the context where you might have in Alzheimer's both tau and both amyloid, you will only visualize the tau pathology. So it's really powerful in terms of differentiating the tau from the amyloid, and also from using that in terms of other kind of tauopathies. And here it's a kind of a combined PET MRI, the MRI structure, and the uptake and the selective binding of this radiopharmaceutical to show the distribution of tau tangles in the brain. So more recently is what we kind of able to look at synaptic density, and you could say very much you can say the synapses and their effect is something that's always been present. You can almost say you can detect it as linked with cerebral metabolism, which we can see in fluorodeoxyglucose. Of course, we do see in terms of the synapses damage in terms of neurotransmitter systems, and we have various PET traces that can look at different neurotransmitter systems like 5-HT1A, which of course it gets lost as the synapses is damaged. But I think the more recent breakthrough which occurs really 4 years ago, is being able to look at the synaptic vesicle glycoprotein, SV2A, which is more prevalent in terms of the synapses, and I think that's always been used for a long time as a in vitro method for assessing the synapses in the brain. And of course, in this case, it's the compound I was developed. Again, this starting point, the carbon-11 use compound UPCJ, and it can pick up the presynaptic vesicles presence, and it can give us a quantitative determination of the presence and the variations of synapses, or this synaptic density. And you can see a good example here, the control, the high presence of this glycoprotein in the brain, well present in all parts of the region. But here in cortical basal, a form of dementia, the real rapid loss or the damage of synapses showing the loss of this SV2 synapse. And in PSP, progressive, you can see even more significant loss compared to that. So it's a really very powerful marker of synaptic density and how it compares to in terms of the pathology. So this kind of thing we can just bring together and apply, and really the end of these things is having them all in terms of the tools to then not just look at them in singular by itself, but to look at them in terms of relationships and of course in time scales. So again, we can look at amyloid with PIB, we can look at neuroinflammation with PK, we can look at the tau pathology all in one patient at different time points, and potentially repeating maybe 2-3 times to see then how they compare to each other in terms of their profile and their relationship. And that's a real powerful thing to understand this context in terms of neurodegenerative disorders. So that's where we are now. So really just kind of show really how can we use it. And I always show this again with a bit of caution, because it kind of applies that this sequence of events actually occurs from A, B, tau, but let's maybe just go with that, maybe accept that. But how can we then use imaging for dementia research? We can measure the changes and progression of the disorder in terms of how, as I just showed just now, the different components and how they vary with time and their relationship. We can actually detect changes in the brain before the symptoms, and that's hopefully we hoping to do before the real changes occurs. So early potential early detection, very much we can say very much in this kind of late early stages. We can hopefully improve the diagnosis of dementia, the different times of front of the dementia from Alzheimer's. So we can then base it on terms of presence of a certain protein, the distribution of that part of the brain, that more characteristic of a disorder compared to that. So that can be a very powerful tool when often symptoms of various dementias do kind of overlap or similar. Of course, the key point is very much help develop new drugs and assess their effects. And of course, one of the things now for many drug trials that targeting amyloid pathology, the entry point is this person actually got amyloid in the first place. So you'll image them with a tau PET to actually see that, and then you can actually then enter as a process and then see the change in that effect. I think I've got time. I might have to speed up a bit. Maybe, of course, in terms of maybe just going to in terms of next generation, is very much then being able to look at various forms of isoforms, and clearly between tau, it exists in 6 different isoforms, and of course different dementias. Alzheimer's has 3 and 4 Rs, while the progressive supranuclear only has 4 R, and some of these PET tracers can pick up different forms. And what we still want to do is work on actually having them more selective, and that would aid in terms of being able to look at the different forms of dementia, Alzheimer's for progressive supranuclei, and really understand this kind of how this maybe the isoforms do make a difference. But one of the things, really things which we are working towards, and the early things, is to move away from to try and get earlier and better detections. Because the nature of what I've shown now in terms of the tau and in terms of the amyloid traces, they really pick up the end stage of these misfolding proteins in the fibrils. What we really would like to do is actually go earlier on into the earlier forms to the oligomers, which are very much formed earlier. And what is perception is that this is actually the more toxic forms compared to the fibrils. This is what it really do the damage, and to be able to image them and be able to kind of prevent them will be a very powerful tool to preventing this kind of their damage in dementia. So we recently got a PRC, a UK grant towards doing that. And what we really want to do, be able to do, is then to develop a PET tracers that can really pick up these oligomers, which tend to be in a smaller percentage compared to fibrils in earlier forms. And what we were able to do is then we came with this compound we call PFTA, which we know actually, if you look at its aggregation profile, you can see the actual, which is based on fluorescence, and it's called using thiaflavin T, which is kind of fluorescent dye, which will fluorescent when it picks up beta sheet containing aggregates. It can actually give a fluorescent. You can see as the aggregation goes with time, with changes from this really along this process, it'll pick up the fibrils, but doesn't pick up the actual stage of oligomers. This compound does, and we can see it picking up very early. So it's a very good point to work on. So we base it on this, but the actual compound was not ideal for the PET tracer in terms that it really didn't have a handle to actually put on a fluorine tracer at all, And also when we found it wasn't selective of oligomers, it picked up both oligomers and fibrils, so it wasn't ideal and it wasn't stable. So we made a variant on this, which incorporates the fluorine itself and gets to that. And we find this, and I'm going to show later very soon, it was certainly more selective towards this oligomers compared to fibrils. And we can see this here in terms of that. So going back to the similar kind of aggregation for which I showed you just then, which again, here it is images of the actual proteins from the monomers to this oligomeric stage, kind of slightly larger, and then these are the kind of fibril stages, which kind of, this is all electron TME images of them. So we can see how they form through this 0 to 60 minutes. And again, based on fluorescence, therefore these compounds will fluorescent when if it binds to any of these forms. You can see the changes. Again, the reference compound thiaflavin, no real uptake until you can pick up this, while a PTFE early stages, and it picks it up, this form very, very nicely, this amyloid, and we get the same thing for tau. So we soon have a compound which I want, in mind of time, show all the data, which has very good selectivity and affinity for the oligomers. So it shows the potential we can really use this to go in earlier forms of tau. And I'll just quickly show some in vitro data showing PSP, AD tissue and AD. And you can see if you do a staining with AT8, which tends to be for kind of the amyloid, tau itself, you can see it correlates with our stain in both cases, but it does not actually correlate with ATA, which tends to be more specific for the fibrils. So showing it, this compound of ours is more selective to the oligomers compared to the fibril forms, which is for both for AD and for PSP. So this is really where we want to go, start going to more earlier forms of able to pick up the earlier forms of the misfolding proteins as consistent with dementia. But I'll kind of come down to, I suppose, going down from very in vitro to I must say, a larger scale. As I said, one of the things I do, apart from the kind of my Cambridge role, is really to, in a sense, underpinning with leading with the Dementia-Platform, which is a UK organization that was established about 6 years ago, really to work on a national level to facilitate and accelerate discovery of new ways to understand diagnosing diseases. And kind of very early on, it was recognized that imaging can be a powerful tool towards that. And one of the things that over the years we've created is to kind of roll out a network of tools for underpinning imaging, MRI, but in particularly in terms of PET, and created for the very first time in the world a network of PET MRs of one group of consortium working together, and it now consists of 8 centers, Edinburgh and KC and UCL, which have Siemens MMR, and Newcastle, Manchester, Cambridge and Imperial, and only more recently this year joining us is Sheffield. And what this does, it actually brings the technology closer to patients. They are embedded next to the clinic, they're next to the memory clinic, so in essence, more powerfully enables the cohorts of patients to get access to these scanners. And this is a unique platform for enhancing the dementia platform wider goals in terms of research into the disorders, but also creates a unique multi-centered platform for clinical trials, for improving a wider range of cohorts, and also we're working with pharmaceutical industry. And it's really something which we are really building on and see unique aspects of really you could say the UK dementia space. So what we've doing so far, and I think it's been early stage, is to really to confirm that in terms of if you do a study in Edinburgh with a patient, you can then match that data with, say, a study in Cambridge or at a UCL. So we are doing a study we call harmonization, where in essence, patients will compare one person, a patient or volunteer will go to Edinburgh, get scanned Edinburgh on their PET MR scanner, go to Cambridge, get scanned on the Cambridge PET scanner, and what we aim to achieve that our data is similar. We can then pool all our data when we start doing multi-center studies, multi-site studies, all into together, because the data can be considered to be comparable, and there's no difference that's linked to the scanner or in any ways in that. And that's what we have at very early days, in fact, using beta amyloid imaging as the tool towards doing that, to intend to harmonize and quantify measures and really can see, does our data vary between scanners and between the way we reach protocols. And once we've done this, really we'll have what it is, a really unique platform of a UK-wide network to really to drive molecular imaging in terms of using it for dementia research. So I'll end there, and I think I'm almost on time, so hope I didn't rush things too much. But just kind of always catch kind of the acknowledgements, for the groups in Cambridge, which is all based here, and wider people which I won't name, but also Dementia-Platform, which it's a UK-wide initiative for centers from all around the UK, really working together, from kind of the clinicians, physicists, chemists, and in terms of application. And then, of course, then finally, and I'll just put a few more funders, of course, the Medical Research Council, which funded that network, EPRC, which funded many of the programs which I've mentioned, the Cambridge Biomedical Imaging Centre, and Wellcome Trust, and a range of course, the charities as my research on that. And then finally, maybe I can, of course, thank you from Rizona for, I'll say attendance, but for joining to this webinar, and hopefully I've given you kind of a flavor and the idea of what and the power of molecular imaging can be applied towards molecular research. Now, I'll stop there. Okay, thank you.
Thank you, sir. Thank you very much, Franklin. And I must confess that I've also learned a lot. I work on Alzheimer's, but very basic, from kind of basic science background. So I didn't really realize that a lot is going on in that PET or imaging areas. When you showed that you're developing tracers for oligomers, etc., I was quite excited to see that. So thank you very much. So to all the audience on Facebook and those on Zoom.
Right, stop sharing.
Yeah, yes, you can stop sharing.
Okay.
Yeah. So you can send in your questions, and I think we've got one question here. It's said, when was PET/MRI first used in combination like this? I think it's probably referring to one of your slides in you.
Yeah, well, we've always used both methodologies for years as separate things. Because we always need the resolution and structural resolution of MRI. So a patient might come in for one day for a PET scan, and then next day for a PET. But more recently, actually, I think it's like 6 years. It first with Siemens with the MMR developing, well, I think that's earlier phones by Philips, but developing this unique combination. So it's really over the last 6-7 years, and I think in Cambridge we installed one less than 4 or 5 years ago. So it's a more recent thing in terms of combination of PET and, you could say, a multimodality imaging. It tends to be CT, has always been, the vast majority of PET scanners are PET CTs, where they use the CT for the structural aspects, not comparable to MRI, but it's still that's always been a combination. And it's really only now, in the last 6, 7, 8 years, has it been PET MRI. It's a recent thing, expensive thing, but it's a recent.
Now that you mentioned expensive, I was wondering, in this imaging, is it always expensive? I mean, for people living in places with less financial kind of boost in areas of diagnosis or research or something, are there cheaper alternatives?
Well, for what the kind of thing I'm showing, I suppose it is expensive. You need that infrastructure. Well, if you really want to have the ability to everything what we do in Cambridge of having a cyclotron and having this kind of everything, then it is expensive. But one, of course, as I show often, is that we're moving away from these very short-lived radioisotopes to making ones which have the fluorine-18. So what's happened, and I think a good example of in the cancer field for cancer diagnosis, is that I have 2 or 3 centers in the UK that will make the pharmaceutical on a larger scale, multi-dose scale, and will ship it to other hospitals. So a hospital will have the scanner. I suppose there's still a requirement for this scanner, which will be about £1 million or so. So it's still a kind of expensive thing in that aspect. But at least it takes out the quite expensive infrastructure of the cyclotron and the radiopharmaceutical. So in terms of can it be cheaper, I suppose the potentially cheaper alternative to PET is a technique called SPECT, which uses far much longer lived, and that's far more prevalent in terms of hospitals and probably in more worldwide hospitals available. But of course, doesn't have the same kind of power and sensitivity as PET, but it can give you, to an extent, some of the information that's obtained from PET imaging, and so that could be the cheaper alternative.
I see. There's this question: how is the other markers that you showed, like the other markers that you've shown for synapse and for the microglia, activated microglia, are they already in clinics? Sorry, I think this is a bit from someone from not in the background.
In terms of, when you say in terms by clinic, in terms that they've been used as
widespread
research, yes, they are. I showed one called Carbon-11 PK295. That's been used since, I think, the late 80s. It's been used at the gun into man. And what happens very much in the last 10, 12 years is that there's a real recognition that inflammation is such a powerful, it's just a key component of dementia. It's not just kind of a potential outcome. So there's been an expansion of various radio tracers that target this receptor called TSPO, which is present in active microglia. But there are other kind of potential targets that could give maybe more nuanced information about neuroinflammation, and they are coming through, and they're all being radiolabeled with fluorine-18, and they are actually going into clinic. In essence, clinic means that they're being used in humans, not necessarily being used as a kind of routine diagnostic tool, but certainly being used routinely on a research level in many places, UK, Europe, and US.
Yeah, I guess the question was referring for clinic. So there's a question that said, considering that PET tracers are radioactive, how safe are they? And which is the maximum number of scans that a patient can go?
Well, previously you could say it. I always used to say, once you say it's similar to someone traveling across the, to Atlantic in cosmic rays in a plane. In terms of the radioactive dose someone will get. Normally, previously most I've had about mostly 3 scans has been a norm. But the one, of course, is in terms of technology, the sensitivity of these scanners have got significantly better. So the amount of activity, the radiation dose you will give to someone to get a kind of usable signal for the brain is going down. So it's just potentially moving from potentially 3 or 4 in one patient. Now, of course, when I say patient, I suppose maybe in dementia, when someone is, let's say, oncology, where they're having radiotherapy, then in the context of this, of a PET tracer is far less, so you could have more. But one of the real exciting breakthroughs really much in the last 2 years is what we call the whole body PET, where instead of just scanning or capturing one part of the body, be the brain, be the chest, we can then scan the whole of the body, and the sensitivity for that has gone 40-fold. So that means if like 30-fold or so in terms of injection, and suddenly the idea you can do multi studies in one patient and still keep the same safe limit, maybe 10 or 12. That could be the future. But right now, 3 to 4 is the general norm as what we give in terms of a normal healthy volunteer to use. Yeah.
Fantastic. So normally in this kind of talks, we ask speakers to tell us something about themselves. Well, I've already introduced you, but just to help people in my career stage or are just above me or younger, something about career, could you share some ideas, tips, or stories to inspire people in my career stage?
Well, I think I'll start kind of question once now. And I would say, never be afraid to try something, suggest something. Because many of the things, I would say, I tend to do. And let's be a fact, for instance, as a BAME person in the UK, there's more of a challenge to promote yourself. And often not necessarily part of the natural part of a network or invited to be a network. It's more of an issue, especially on European levels. It's not that. And the reality is often in the sense that many of the things I've just said, why don't we do this? And you kind of find people say this, you know what? I think that's a good idea. Why don't we do that? So I would say don't be afraid to, in terms of proposing something scientifically as a new idea, applying for a grant, upon that. Don't kind of, you always say, people use, as it's a cliche, the worst question you don't ask when you don't ask. I would say one of the things you do is you propose to do something. And I think even more so for being a BAME person in terms of this, because you won't necessarily be invited or introduced or brought into the network in terms of your career. You've got to, in a sense, I would say, put yourself in there.
Thank you very much. This is quite thoughtful, and I felt like you were telling it to me. So
yeah,
thank you.
So don't, absolutely. So you do that.
Absolutely. Thank you very much. I think since now we haven't got any more questions,
Yeah.
I would like to use the opportunity to thank you for honoring the invitation to give this talk. It's really fantastic. And hopefully in the future, you'd come and give other talks, not necessarily about your research, but just about career as a BAME person, etc. Because like you mentioned, as a BAME, I think we really need role models, and we need people to tell us that you can actually do this, you can do this, don't be afraid or don't be scared to do this. Yeah. So hopefully at some point when invited, you can come and share some thoughts and ideas.
It'd be a pleasure to, absolutely. This is one of the things I want more like me in Cambridge and you in Cambridge, absolutely.
Thank you. All right. Thanks, everyone. Bye-bye.
Okay. Thank you, everyone. Thanks, man.
All right.
Have a best.
Thank you.
Bye.